Translation involves decoding a messenger RNA and using its information to build a polypeptide, or a chain of amino acids. For most purposes, a polypeptide is basically just a protein. But first, to understand translation, we are going to look at transcription. Transcription is the process of copying a gene's DNA sequence into a complementary mRNA molecule. Occurring in the nucleus, it is needed as DNA cannot leave the nucleus, so the information must be copied into mRNA, which can travel to the ribosome. mRNA is transported through the nuclear pores into the cytoplasm. Once the mature mRNA enters the cytoplasm, it travels to a ribosome, the cell's protein factory. This symbolizes the exit from transcription into translation. The mRNA sequence is read in groups of 3 bases, called codons. In order for initiation or translation to start, a ribosome which comes in a small subunit and large subunit will need to be present. The small subunit attaches to the mRNA. The ribosome then finds the start codon, AUG. AUG signals the start of translation. But first, we need to understand what the large subunit involves. The A site is the entry point for incoming tRNA molecule, testing if the tRNA's anticodon matches the mRNA codon. If it's a match, the tRNA stays, carrying its new specific amino acid into the ribosome. The P site is the middle pocket, holding the tRNA that is physically attached to the growing peptide chain. During the reaction, the entire protein chain is transferred from the P site tRNA to the amino acid on the A site tRNA. The E site is the final stop for empty tRNAs. Once a tRNA has given up its amino acid chain to the A site, the ribosome shifts over. The uncharged tRNA moves into the E site and is then released back into the cytoplasm to be reloaded with a new amino acid. The large subunit attaches. The ribosome is fully assembled and translation can begin properly. Transfer RNA or tRNA is a small RNA molecule that carries specific amino acids to the ribosome during translation. Its role is to act as a translator between the language of nucleotides, which are codons on mRNA, and the language of amino acids, proteins. tRNA produces an anticodon, complementary to an mRNA codon, ensuring the correct amino acid is added to the growing protein. For example, AUG codes for UAC, producing the amino acid methionine. As an overview, initiation goes like this. First, the tRNA carrying methionine attaches to the small ribosomal unit. Together they bind to the 5' end of the mRNA by recognizing the 5-GTP cap added during the process in the nucleus. They then walk along the mRNA into the 3 direction, stopping when they reach the start codon. Often, but not always, the first AUG. Elongation, or the middle stage of translation, is when the polypeptide chain gets longer. Our first methionine carrying tRNA starts out in the middle slot of the ribosome, called the P site. Next to it, a fresh codon is exposed in another slot, called the A site. The A site will be the landing site for the next tRNA, whose anticodon is a perfect match for the exposed codon, GUA coding for valine. Once the matching tRNA has landed in the A site, it's time for the action. That is the formation of the peptide bond that connects one amino acid to another. This step transfers the methionine from the first tRNA into the amino acid of the second tRNA in the A site. Now we have 2 amino acids and a very tiny polypeptide. The methionine forms the N terminus of the polypeptide, and the other amino acid is the C terminus. But how does the chain continue to grow? Once the peptide bond is formed, the mRNA is pulled onward through the ribosome by exactly 1 codon. The shift allows the first empty tRNA to drift out via the E site, also exposing a new codon in the A site, so the whole cycle can repeat. That covers the elongation stage. Translation ends in a process called termination. Termination occurs when a stop codon in the mRNA, such as UAA, enters the A site. Stop codons are recognized by proteins called release factors, which fit into the P site. Release factors mess with the enzyme that normally forms peptide bonds. They make it add water molecule to the last amino acid of the chain. This reaction separates the chain from the tRNA and the newly made protein is released. Overall, the process of translation occurs in the ribosomes. It uses mRNA codons as a template for coding for a sequence of amino acids in a polypeptide chain. tRNA anticodons carry the appropriate amino acid into place. Initiation, elongation, and termination are all stages within translation. The correct matching of the codon on the mRNA to the anticodon on the tRNA is critical for the correct sequence of amino acids in the polypeptide chain. Polypeptide synthesis provides the means for the inherited qualities that are carried in the DNA to be mobilized into cellular structure and function. This concludes my model of translation and polypeptide synthesis.